Hello friends, please subscribe to my channel Bosons Botany to get more videos, and do not forget to hit the bell icon to get notifications and to not miss any new videos and updates. Thank you and stay tuned. Hello guys, welcome to Bosons Botany. I am starting with structure of the RNA. First I will discuss the two-dimensional model of tRNA, which is also known as the cloverleaf model of tRNA, and this was first studied for East alanine tRNA. So as you can see the structure here, it is quite simple and you can easily understand the structure. So towards this side, that is towards the left hand side, this is the 5' end, and the 5' end will always have guanine as its first nitrogenous base. Rest you can write any kind of nitrogenous base, but the 5' end will always have guanine as its first nitrogenous base. Towards the right hand side, if you see here, this is the 3' end and ACC, that is adenine, cytosine and cytosine. This is the sequence that is followed towards the 3' end. So this is something you need to remember. This never changes. This is similar for every species, for every organism. It could be eukaryotic, it could be prokaryotic, anything. This sequence is always the same. That is, towards the 5' end, the starting nitrogenous base is always guanine, and for the 3' end, the starting nitrogenous bases are adenine, cytosine and cytosine. And as I have already mentioned here, this structure is basically for East alanine tRNA, or you can say for alanine amino acid. So towards the 3' end, this is the acceptor arm where the amino acids get attached, and the first amino acid that will attach towards the 3' end or on the acceptor arm is alanine amino acid. Now if you observe the rest of the parts, you can see this is the T-Cy-C loop or the T-arm or the ribosome binding loop. This loop here towards the right hand side, this is known as the T-Cy-C loop or the T-arm or the ribosome binding loop, which is having 7 base pairs. Total 7 base pairs are present to make this particular loop, and the ribosome binds here towards this loop. That is why it is also known as the ribosome binding loop. This is the lump, this outgrowth here, this small outgrowth here, this is known as the lump or the variable loop or the extra arm. Towards this side, you can see anticodon loop, which is having 7 base pairs, and this anticodon loop is basically having the anticodons, the triplet anticodons, which will pair up with the triplet codons found on this mRNA strand during translation, that is during protein biosynthesis. So the triplet codon which is present on the mRNA strand will complement with anticodon or the triplet anticodons which is present on the anticodon loop of tRNA, and when the sequence match, when the anticodon and the codon sequence will match, then the translation will start or formation of a polypeptide chain will start to generate a protein. So this is the anticodon loop having anticodons comprising of 7 base pairs. Towards the left hand side, this is the DHU loop or the amino acyl tRNA synthetase binding loop, and it is having 8 to 12 base pairs. So here the amino acyl tRNA synthesis binding loop, which means amino acyl synthetase, which is the enzyme to make alanine amino acid, will bind here. That is on the DHU loop, which is also known as the amino acyl tRNA synthetase binding loop, having 8 to 12 base pairs. Now some points regarding the 3D model of tRNA. But before I give you the points, I would like to mention all the points here. The abbreviated the labelings that I have drawn, sorry, the abbreviated letters or the abbreviated words that I have labeled. So DHU stands for dihydrouridine, T stands for ribothymidine, Psi stands for pseudouridine, and C stands for cytidine. So these are the actual names that belongs to all these abbreviations that I have written here. Now the points regarding tRNA 2D model. This was first of all deciphered, or R. Holy was the first scientist to actually give the concept of 2D model of tRNA. So by R. Holy, while working on yeast for the structure of tRNA, found the cloverleaf model for an alanine amino acid. R. Holy, while working on yeast for the structure of tRNA, found cloverleaf model for alanine amino acid. So R. Holy was the first one to give this model. R. Holy, along with Nirenberg and Herwin Khorana, deciphered the nucleotide pairs. tRNA has 77 to 80 genes, and this is for yeast. For yeast tRNA, there are total 77 to 80 genes. In between this bandwidth, 77 to 80 genes, and this was first of all given by Herwin Khorana, for which he got the Nobel Prize. So the contribution of Herwin Khorana is very significant when it comes to gene or genetic codons or deciphering genetic codes. His contribution is extraordinarily massive. So you need to remember his name, his contributions, and for what kind of contributions he actually got the Nobel Prize. So for this also he was awarded the Nobel Prize for deciphering that in yeast tRNA there are a total of 77 to 80 genes. Now if I give you this 3D model of tRNA, which is quite similar to the 2D model of tRNA, the things are similar, but there is a very small difference. So as you can see, again there are 3 lobes or loops, whatever you want to say. The first loop here, it is T-Cy loop. This is the 5' end having guanine as its first nitrogenous base, and this is the 3' end having CCA as its first nitrogenous bases. This is the T-Cy loop. This is the DHU loop or tRNA synthetase binding loop. This is the anticodon loop, and the length from this point towards the end is 77 Armstrong. And this is the 3D model of tRNA. Some points regarding this. Klug, with the help of X-ray crystallography, studied many 3D structure of tRNA and got the Nobel Prize. So Klug, just for studying the 3D models of tRNA using X-ray crystallography, he got the Nobel Prize. Later on, Kim finally gave the 3D model of tRNA, which is now very popularly known as inverted L-shaped tRNA 2D model. As you can see, this is inverted L-shaped model, and this was given by Kim after several contributions of Klug. Kim got to know the 3D model of tRNA, which was supposed to be like an inverted L letter. So this is the 3D model of tRNA, and this is the 2D model of tRNA. So very often you are asked who are the scientists who gave their contribution for establishing the structure of tRNA. So first, if you observe this is the first structure that we study, and after that we study the 3D model. So for the 2D model, R. Holy was the pioneer scientist, and later on Nirenberg and Korana, they gave their contributions. And for the 3D model, which came a bit later, this was given by Kim, but with the help of Glug. So this is all about tRNA structure. So thank you.